a lot of the things that I work on. So on the left here, these huge sort of big piles of cobble and stone are all left over from hydraulic mining back from the gold rush era. And this is a river that's today, you can still see the remnants of hydraulic mining. On the right, you can see a recent wildfire burned through this part of the watershed. And there's all kinds of interesting things. And on top of that, this river is dammed and managed for hydropower. So there's a lot of things that are happening. And so the critters that are living in the river are still there, which is pretty cool. And so today I'm going to talk a lot about how I use a bunch of different tools, largely genetic tools nowadays, to learn about the stuff that is happening in these streams and rivers and some wetlands too. So let's get going here. And so I first want to just sort of give folks a chance to think about what your favorite river is. I'm going to talk a lot about rivers and wetlands, but there's been studies that have shown where people go to a place with flowing water and it actually reduces your blood pressure. It brings health to you physically in a way that not much else does. And so I think it's nice to sort of think about those places that you've been, and maybe if you have a favorite, you can throw it in the chat if you're savvy with that. I'd love to know what your favorite river is that you would claim. Mine, I would say, are these two. The one on the left here is the North Fork American River, up off of Highway 80 near Colfax. It's one of the only undammed stretches of Sierra River and left in the Sierras. It's beautiful. There's lots of frogs and all kinds of cool fish and stuff in there. It's great. And then I've done a lot of work in the Southwest, actually. And so this is actually a picture of the Yampa River, which is a tributary to the Colorado. I've done work in the Grand Canyon, which is amazing. But the Yampa River is just a really cool, amazing landscape. And I really, really think it's one of my faves. So I'd put those two down. So think about that. And then I want to read this quote because I think I think it's very impactful for many reasons, but it's by Mark Twain. And the face of the water in time became a wonderful book, a book that was a dead language to the uneducated passenger, but which told its mind to me without reserve. And it was not a book to be read once and thrown aside, for it had a new story to tell every day. And it's actually referring to steamboat captains and the fact that they had to memorize the river and all the things that the river changed when different floods came through, because they needed to know where to go to basically drive their steamboats. But I think it's really applicable to a lot of the things that I study and that we are still trying to figure out. But rivers have lots of stories to tell. Wetlands have lots of stories to tell. And sometimes it's just figuring out the best way to get at that story. It's almost like trying to solve a mystery, and we don't really know. We don't have all the pieces, and we're trying to figure out what are the pieces we need to figure out a solution. So my background, as Brooke mentioned, I've done a lot of different stuff. I'm a rafting guide. I've done the Grand Canyon a bunch of times. I've been lucky enough to do it for a class to help teach folks. I've worked for the National Park Service up in Sequoia and Kings Canyon. I've worked for consulting firms. I've worked for the Forest Service, and now I'm at UC Davis. And so I've had a lot of opportunity to work in all these different places, which has really been a wonderful way to sort of learn different perspectives and viewpoints, which has really informed a lot of the work I do now. And I like to start with my acknowledgements up front, just because sometimes I get shoved at the end. That's my son from many years ago, actually. Their kids are great frog catchers. So a lot of folks are going to be really important in the talk I'm about to give. And so lots and lots of people deserve a lot of credit for both the funding, help, hours in the lab, and otherwise that I'm going to mention. All right, so let's get into the fun stuff, which is freshwater systems at risk. So it's probably not a mystery to y'all that there's a lot of things going on with our freshwater systems. There's major threats globally to biodiversity from, these are sort of I would say the 5 main things that are often listed, but there's also climate change is obviously a big important piece of this, and I would say it's sort of the Death Star that links all these things together at the moment. And one of the problems, or things that we face, is that we live near the water too. We need it, and so over half of the world's population lives within 20 kilometers, about 10 miles of a permanent river. So there's going to be, some sort of reckoning where we need to figure out a way to balance our needs and uses with, the native freshwater species that have lived there. And that means that there's always going to be sort of this interesting sort of juxtaposition or conflict between human water security, which is up on the upper right panel there. You can see actually it's a map. So the hotter the color, it means there's more risk to our water security. And that could be clean water, access to water, all kinds of different things, right? And then on the bottom, it's threats to biodiversity. So all the plants and animals and things that use the water as well. And you can see those maps kind of look identical, right? Like, not surprisingly, the places that are most at risk are at risk for us and for everything else. And there's a great book I want to point out. It's a fictional book. It's a fun read, but it's called The Water Knife. And I had a chance to actually go down the Grand Canyon with the author, Paolo Bacigalupi. But it's sort of an interesting futuristic take on water in California, Arizona, and Nevada, and sort of the drought really extends into the future, and we're right in the thick of it right now. So there's a lot of things that hit fairly true to form there. One other thing I want to mention, I'm talking more about this, but we have changed the landscape dramatically, especially in California, more than most anywhere else, really. To the point that this quote I really like, because we acted upon the Western landscape with the force of a geological agent, because we've made changes to our landscape that are so dramatic. They're greater than or equal to like what millions of years of geology has done. And so now we are sort of trying to remanage or replumb our system in a way that it did not evolve to do. And I love this far side because it's sort of like, hey, we're these little fish in this globe, and we may have broken some things, and now we're trying to figure out how to fix it. So what? How do we monitor and assess changes? How do we learn and manage our systems, our freshwater systems, so that we can both enjoy them from a recreational standpoint, whether it's rafting or fishing or just going out and going for a walk? And then how can we also consider, the things that we also use them for, hydropower, right? Or agriculture, any of those things. And so I'm going to talk a little about some of the different approaches that scientists often use in this regard. Is observational. We go out in the field and we count how many frogs or fish are there. Or we use experiments in labs, right, where we catch some stuff and then we measure it and test how things react to different chemicals, whatever it might be. And then finally, there's sort of a third approach called synthesis, which is where you take a bunch of existing data that's already been collected and then you kind of figure out how to mush it together and understand something additional from it that maybe others hadn't done. And that's largely sort of in the realm of data science, right? So you're sort of analyzing data. And I would say that where I have fallen, I've done all of these, but what I've ended up using actually is genomics, which is sort of the study of genetics or using genetic data, because you can do quite a few of these things. You can even use it experimentally. I don't do that, but folks do. And it's a pretty amazing tool. It is one tool. There's lots of different tools we have, but I'm going to talk about how it's a really exceptional tool for using in a bunch of different ways. So genetic tools for conservation. One of the big problems is, a lot of the species that I study and that folks are often interested in are not well studied, right? We don't know much about them. And on top of that, they're really cryptic. They're hard to find. Frogs are really tricky to find. Often they live in really amazing, beautiful places that are also very inaccessible. So you're sort of adding layers of, it takes a lot of effort to go out and catch one frog, or even, if you're thinking about fish, similarly, there's some places where it's very hard to go out and like collect information about the thing that you're studying. And then on top of that, a lot of the declines that we're seeing globally, but especially in the US, are occurring in protected areas. So even in the national parks, which we would consider some of the most well-protected lands in the US, they're still seeing really big declines in sensitive, or endemic endangered species. So protecting the habitat isn't just that's not the end of the story. That's not going to help us. But what's really cool is the tools to obtain genetic data are continuing to improve to the point, I mean, it's almost science fiction now what you can sequence. I'll talk more about that later, but the cost to do this has dropped dramatically and it continues to drop. So this is just the cost of sequencing per megabase. So however many thousand bases, right? In DNA, we have 4 base pairs, A, G, T, C. So 1,000 of them, and this is sort of the cost as it through time as it drops, and it's continued to drop. I need to update this plot. So why is this important? What does genetics tell us that other tools may not? Well, from a long-term perspective, genetics is really the source code. It's the material for future adaptation, or the adaptive potential of everything, right? So it's the kind of it's the final thing that determines how things can or may adapt in the future. And in particular, in a lot of places where we have really small populations, whatever that might be, birds, frogs, fish, plants, when they're not able to connect with one another, they often have much less genetic diversity because they're not sort of passing their genes back and forth, which means they have reduced adaptive potential. And another way to think about that is just genetic health, right? Like we don't have much genetic diversity. That's not generally a good sign because that means one disease could come through and wipe a whole population or a whole species out, or even one sort of catastrophic flood or event that's just a random event. And so the genetic health of the population is really this sort of really crucial metric that we can measure, and it's proportional to the ability for that organism or population to respond to current and future environmental change, right? And so with climate change impacts ongoing, this becomes a really, really important thing. Like as we have more droughts and more in California in particular, we're going to experience more droughts. We're going to experience more dramatic storms. So this sort of whiplash where we're going to go from really dry drought years to winter storms is predicted and is coming. They're showing that this is the case. Warming temperatures, all these things, they are happening at a very rapid rate. And so the populations that have a good amount of genetic health might be able to weather that storm, so to speak. And so when we're trying to think about applying this, how do we use these genetic tools? Well, what's cool is we currently largely, most of the genetics has been used for really determining populations. And phylogeographic structure means sort of where things are on the landscape. Like, these frogs and these frogs are related to each other, but they're really distinct groups, right, genetically. And so we might want to protect them independently of one another. And salmon is a good example of this. We've used for different sort of fall run salmon, winter run salmon, have different specific signatures, and they want to kind of keep those. And so we can also use it to estimate genetic health, bottlenecks, the actual size of the population, all kinds of cool stuff. And even now, even more, we can actually identify the genes, specifically genes that are the ones that are doing the adaptation, right? So maybe a gene that makes, that puts on fat. For example, they've identified what they call the tasty gene in salmon, right? That makes a lot of fat. And so the salmon that have this gene and it regulates a lot of putting on fat, turns out like it's also correlated with fatter, more delicious salmon. So all these things are really interesting and we can understand them using genetic tools. All right. And then we use those to prioritize what we do, right? Like how much money we put into conservation of habitat or, reintroducing a species or whatever it might be. And then I would say the thing that I spend a lot of time on in particular is genetics really helps us draw boundaries in a way that if I were to go out into a river and maybe I counted 3 frogs and I went to another spot and I didn't count any, that doesn't necessarily mean they weren't there. So it's really hard to draw a boundary and say like, well, they're not here, like we're excluding them, their habitat is no good. Genetics is a better way to do that because it sort of allows us to have a better sense of how to delineate those boundaries in a way that we can manage. And so this is a map of California, and it's a map actually of the range of the foothill yellow-legged frog, which I'll talk about more later. But what's interesting is it's sort of broken into these different groups. These are where these white lines encompass, right? And we wanted to figure out, like, okay, are these genetically distinct or not? And so both myself for my dissertation, my PhD, and another researcher sort of independently did some work. And what was cool is we both came to the same conclusion independently, which was great. And we used those, so now these dots are colored, and we use those sort of different colors here as how to recommend that these frogs would be managed. And what's really neat is that went directly into the endangered species listing for the state, which has been accepted recently. And now the federal listing is actually using that same data. So it's been really neat to see sort of research go directly into management in a way that's useful, because we can now prioritize. We know on the North Coast, for example, there's lots of frogs. They seem to be doing pretty well. Genetic health is good. And in the Sierras, and especially in the South Coast, they're really not doing well at all. Like there's literally one population, two populations left. They're probably going to go extinct down here. So this is like a very, very high priority, and genetically that we can see that as well. Okay, so how do we do that? Like, cool. So I'm going to try to explain what genetics is nowadays, actually, which is kind of a cool process and still a little mind blowing. So I work with frogs, but I'm going to show an example of some other. I use fish. I use these little zooplankton, all kinds of different things. So you can do it with anything you want. But the first step is you go out and you need to collect DNA. For fish, sometimes we'll take a scale. For frogs, we actually swab the mouth. It's like crime scene investigation. We go in, we kind of swab the mouth. They're not super happy about it, but the great thing is it's non-invasive. We can let them go and they're fine, right? They've just basically brushed their teeth. They don't have any teeth, but anyway, you get the point. So we swab their mouth, and then we take that swab and we stick it in one of these. I don't know if you all can see my mouse, my little pointer. Hopefully you can. But we put it in one of these little vials, right? And that little kind of murky liquid that's in that picture, that's actually the DNA that's been sort of, the cells have been lysed. So they break them apart. We pull the DNA out and we make this slurry. And we do that for every individual that we catch. So for example, I catch a frog, swab it, put it in a vial. And then that all goes into a 96 well plate. So I don't know if you can see it in here, but at 96, so now we have 96 frogs, and they all have their own individual DNA in one of these little vials, right? Or we call them wells. So it's 96 wells on a plate. What's crazy is then all these little things that are sticking down, these little straws, are pipettes, and we use a 96 well pipette machine. Basically, it's just chunk, and you just pull it down, and then we essentially take all that DNA, and then we add barcodes to it. And when I say a barcode, I mean a genetic barcode. So we actually have individual AGTC combinations that we make in a little vial, and you drop a little bit of each of those into one of these wells, and it individually codes all of those samples. So now I have 96 frog samples. They each have their own little barcode, genetic barcode, right? And then here's the crazy part. So then we take all 96 of those samples, because they're individually barcoded with DNA on either end of sort of imagine DNA is a string, right? It's a strand, and we've added barcodes to either end of that DNA. And this is all happening sort of magically in that vial. Then we take all of that and we stick it all into a single one more vial. We take a whole 96 well plate, 96 samples, and we put it into one single vial. And that vial then gets its own another barcode. So all 96 samples now have an additional barcode that is for the plate. So I can now combine 96 samples at a time into a plate. So maybe I have 10 plates. So I have almost 1,000 samples, and I collapse them all down. And then I can put them all into one single vial. So that little tiny dot, that little black speck there, represents over 1,000 individual frogs that have been barcoded and are now literally condensed. All the DNA is just in that tiny little black dot. I walk that across, which this part is the crazy, scary part, because you're like, this is my whole life in a tiny single vial. I walk it across to a sequencing center, which I work at UC Davis. We're lucky we have one on campus. It's like right across the hall from one of our labs. I walk it across and they basically stick it in this big machine. And then they send me just ridiculous amounts of data on a computer. And then I spend the next however many years processing all that data. I'll talk more about that, but that's genetics, right? So that's how this works now. The lab process itself is actually quite fast. And the reality is that it's generally all the back end of sort of pulling all that data out. Because then what I can do is I can take those barcodes that we know and I can sort of extract everything back out based on those barcodes by the plate and by the individual. And then I have individual samples that I can work on. Cool, everybody with me so far? It's kind of science fiction, I know. All right. And I want to show this, it's kind of cool. So we use a lot of these little magic magnetic beads. DNA is negatively charged, so we can leverage that with sort of these nano beads that are magnetically charged as well. And so what you're going to see is a little video, hopefully. Brooke or somebody give me a thumbs up if this works. But this silver thing is a magnet. And you'll see little brown little gunk, which is the DNA, and it has beads on it. And when we're sort of washing it, we use these magnets and we just turn the vial. You'll see, so it's stuck on one side and see it just shoots across inside all the way to the magnet. And that's basically how we wash or sort of aggregate that DNA. So I don't know if you all saw, I'll play it one more time. So watch the brown stuff kind of shoot across and stick to the magnetic side of that vial. And this is a very common method now that's used because it's very clean. And then the nice thing is you can actually hold that vial and just dump out the liquid and all the DNA is stuck to the inside of the vial. We don't lose any, which is also kind of crazy. So anyway, there's like I'm in my office and there's a lot of students outside yelling and screaming. So hopefully you all don't hear that. Anyway, so that's how we purify DNA, which is kind of crazy. All right. So let's talk about how we use this information, right? This data. And hang on one second. I'm going to thank you. Thanks for that interlude. So I'm going to talk about zooplankton as an example of some wetland research that I'm using this data for. And so this is really cool. I'm excited. This is a work with Caltrout, which has been really fun. And so zooplankton, what are zooplankton? Well, they're floodplain food for salmon and a lot of other things. But essentially, imagine this is your wetland. We've got a bunch of cool little green plants here. And they, all the bacteria and all the little microorganisms that are in there feeding on the green things, right? So the plants that photosynthesize, phytoplankton and algae, similarly, zooplankton are feeding on that. So they're passing that energy up. And then fish, in this case, little baby salmon, love a lot of the zooplankton or invertebrates. And what's really interesting is we've done a lot of work. This is up in the Sacramento area on the YOLO bypass. If any of you are familiar with that, it's this big floodplain or artificial floodplain, essentially, that sort of is just outside Sacramento. And we've done sampling when that area is flooded. And so this is a water sample from the bypass on a floodplain. And all that murky junk in there is zooplankton. It is chock full. I mean, it's just ridiculous. And then in the river, at the very same time, if you sample, you'll see it's pretty clear. There's not much food in there. And so historically, fish used to prefer to go out onto the floodplain, feed, get really fat and happy, and then go out and migrate into the ocean, do their thing, and come back. Unfortunately, that connection in many places has been severed, right? Like there's no floodplain. Well, there's not much floodplain left. And the few places that it does exist is very hard for fish to get to. And we've measured the size of fish. These are fish that we put in cages in the in the habitat. And we went out and measured every week how they grow. So they came from a hatchery. So they were all the same size at the start of the experiment. And this is about 6 weeks in. All the fish that were in a floodplain cage, these are the same age fish. I promise. There's no tricks in here. This fish is 6 weeks old. It was living in a floodplain cage. This fish is 6 weeks old. It was living in a river cage. And we've also, looked at the actual stomach contents. And a single salmon from the floodplain had 1,200 individual little zooplankton, little cladocerans in its stomach, which is crazy, right? So these zooplankton are super, super nutritious. They're like the Snickers bar of salmon. And so Daphnia pulex, which I'm going to talk about, is one of these little zooplankton. Look at this cute little guy with his little antennae. They're like Snickers bars for salmon because they're really lipid rich. So they have a lot of really high fat content, which is great. They're a good indicator of floodplain habitat because they're terrible swimmers. They just sort of float around. They can't really swim. Look at them. They're just little cute little buttons. They're really easy to sample because they don't swim. So we just literally go scoop up with a net and we can count however many are there. And the other cool thing is they're really well studied. So they're one of the first crustaceans to really have a full genome sequenced. So there's actually a lot of genetic information already available. And so one of the things that I've been working on is making something called a genoscape, which is kind of mapping where these little critters are and using them really as kind of like fast track of food for salmon, right? So we can track connectivity of this important food resource using genetics. So we can actually take the genetics of each individual and then look at like where did they come from and where are they connecting. And so this is a sort of a stylized map of Sacramento is over here. This is the American River coming in, and this is the Feather and the Sacramento up here and the Cosumnes. And so the water sort of naturally flows down and then out into the bay, right? And the thing is, we don't know really where some of these zooplankton are moving, how far they're moving, where they're going, and over what time periods. And so we did a little pilot study, which I'm going to show you the results of. And what's cool is, so I took samples from each of these orange dots on the left. This is the Butte Sink, which is kind of a floodplain area. And then the river flows down to Colusa, and we took samples from near Colusa. And then the yellow here is the bypass. So it is a floodplain, and we took samples from there. And then we also were up in the Cosumnes River, which is one of the only natural floodplains left, like in the entire western sort of Sierran area. And so we looked at all these, right? And so we can, the arrows show like where the water is flowing. So it flows out and then into the bay and out, right? And then on the right, this is something called a PCA, or a way to sort of, when we take all that data, those thousands to millions of base pairs, and we actually sort of collapse them into a single point. And we do that by essentially saying like, how different is this sequence from another sequence? And then we kind of measure those differences, and then we use that. So each of these points is a single sample. So in this case, each of these points, whether it's a square, circle, whatever, is a individual zooplankton, or Daphnia pulex, right? These little water fleas. And each one is color-coded by where it's from. And so what's really neat is when we look at this, we can use this so you can see any dots on here. The closer they are, the more genetically alike they are. And the farther apart they are, the more genetically distinct they are. And so these PCAs are a really great way to look at the data and tell us about, like, are the populations really different? Are they mixed up? All these types of things. And so we can see Butte, which is the one up here, right, is you can see, you can draw a circle around it. It's pretty distinct. You can see the population's pretty distinct. And then similarly, Nags, which is our floodplain, is also really distinct. And these two don't really overlap, right, which is kind of cool. So, you know, unsurprisingly, these are not able to swim all the way back up here. But it also means that, hey, maybe these don't actually make it all the way down here, right? Because you might expect to see some more of these kind of mixed in this same space, and we don't. They're very separate. However, we do see this big purple blob here, right? Which is from samples in between, which is cool. So we are seeing genetically the signatures are sort of overlapping those two things, which makes sense. And then finally, what's really interesting is the Casumnus, which is a very different, watershed, and it doesn't connect with these at all, is very, very distinct. And so the next steps of this are we're going to go sample down here and in a few other spots along this, and we're going to see like, hey, we might be able to say from the samples we get down here, we might be able to say, oh, they actually came from this spot, or they came from this spot, or maybe, who knows, I doubt it, but maybe they came from all the way up here, right? And that helps us actually inform decisions about like how water is moved around and how we can sort of leverage this to essentially make hopefully some better food situations for the salmon who already have a very tough time. Yeah, so that is sort of one approach, one ability that genetics is telling us something that would be very hard to figure out otherwise. All right, so I'm going to move now to frogs, which is one of my faves. My kids, I love it. They're just, anytime they see or hear about frogs, they're always pointing it out to me, which is great. I feel like I've succeeded. So frog conservation. I want to talk a little bit about frogs just in history and why they're important, not just to me, but to a lot of things and a lot of people. So in Egypt, this is a hieroglyphic from like 3,000 plus years ago. And that's a tadpole on top of something called a Shen ring, which in hieroglyphics means eternal repeating life or infinity. And so tadpoles and frogs were used to represent eternal repeating life, which is really, really cool. And they also represented the number 100,000 because, anybody that's seen lots and lots of tadpoles knows that, like, it's hard to count, right? Just it means 100,000. Great. And the other really cool thing is in a lot of Native American cultures, frogs represented the ability to travel between two worlds, right? Or communicate with all beings in both worlds and serve as mediators. And so I find this really cool because I feel like I'm kind of constantly trying to drag people to use or translate genetics for people that are biologists or managers or, people that have no background in that. So trying to sort of be the mediator between worlds. And I really like that. And, frogs and amphibians in general have been symbols of good luck, rain, renewal, as well as bad luck and disease, right? So they sort of have represented many different things through history. And I think most importantly, what I think is really fascinating is that they really link the aquatic ecosystems with the terrestrial ecosystems in a way that nothing else really does. And what's really neat is they're actually transferring energy, right? Carbon, if you think about it, like at its very basic level, tadpoles are eating algae, and then those tadpoles, so in the stream, so they're pulling energy from the stream. And then when they metamorphose into adults, they are basically then transferring that, that energy out to wherever that adult frog goes. And, birds eat the frog, snakes eat the frog, like all kinds of things that are more terrestrial. And so that energy sort of flow is being pulled out of the river and the streams and the wetlands, right? Which is really cool. And, amphibians have been around a long time. They're one of the oldest lineages of organisms. Unfortunately, they are also the most at risk right now. So of any taxa, they're at the greatest risk of extinction globally. So pretty important to figure out what's going on and see what we can learn from them, because they really are in many ways sort of a good indicator of how things are doing in the ecosystem. Okay, so I'm going to give another example of a cool project I'm doing in the Southwest with relict leopard frogs. And these guys are really interesting. So they hunt during the night and sleep during the day. I like this little infographic from the National Park Service. They live, around 4 to 5 years, we think. And they are pretty much like a hot springs frog or a springs frog. They live in really cool little side canyons to the Grand Canyon, to the Colorado. And unfortunately, they were one of the first amphibians in North America that folks thought had gone extinct. And it wasn't until they found a couple populations that were just hanging on that they decided, hey, we're going to take a bunch of these frogs and we're going to take them into like a zoo facility and we're going to rear them there, sort of keep them there, rear them. And then we're going to reintroduce them out into the wild in a bunch of different places, some where they historically lived and some places that are new. They, we don't have any records of frogs being there, but they restored the habitat and they're like, let's get them out on the landscape. And so one of the cool projects we could do is essentially we take the samples, they have samples from when the populations crashed in the early 90s, mid 90s. And then they have samples from just recently, like 2018, 2019, when they reintroduced these populations out into the wild. And we can compare the genetics between these to see how genetic diversity changed, right? Did we lose, how much did we lose? Are there populations that are actually gaining diversity? Are there sort of all kinds of interesting things we can learn about? And so what's really neat is these, I had a chance to go out and visit a bunch of these sites. These dots are sort of, sorry, this map isn't the best resolution, but really these come from two core populations. So these sort of this little group of dots here near Lake Mead, and then there's a little group up here. And then all the purple and yellow dots that are kind of sprinkled are all sort of reintroduced, right? Or translocated is another way to say, where they've sort of been reared and moved elsewhere. And so we looked at that. I took these samples and we ran the genetics. I got to go out and visit and do some sampling. This is right below Hoover Dam. We had to have like security clearances to get in here. It was pretty fun. There's a little hot springs that these frogs hang out in. They're not there anymore. Unfortunately, this invasive snail got in there and kind of wrecked everything. But they're in these really cool little side canyons. We got to drive out into some of these places that, it's like Indiana Jones territory. It's great. And they live in these just really beautiful little side oases, basically, including some right around Lake Mead. And this is called Blue Springs, which you can drive. There's a road and this is the picnic area right there. And there's like African cichlids that people sort of didn't want in their aquarium, and they drop in this little pool. And so there's frogs hanging out, and then there's these like totally introduced cichlid fish, tropical fish in there. It's really fun little spot. So anyway, we looked at these different areas, and I'm going to show you kind of a crazy plot and explain it. Don't worry too much about it. But basically what I want to point out is that the colors of the donor area. So we had two main donor areas. Green was the North Shore, so one of the areas, and then the other one is Black Canyon, I think. And so we had each of these boxes is a different place where they reintroduced, a bunch of eggs or tadpoles or frogs into the habitat over time, right? So it started in like the early 2000s, and they kept doing it year after year after year. The number of animals released is the size of the dot. So places like Grapevine Spring, they used, in this case, 10 egg masses and released over 750 individual tadpoles, which is a lot. And they did that through time, lots of individuals. The only thing I really want to point out is that generally they always kept these two donor groups separate, right? Whenever we introduced frogs into a place, they said, hey, these are all from one population that's genetically distinct. Because, there's good reason to do that. We don't want to have some of the things that can happen with when you mix genetics up in a bad way. But one place they actually did mix it up. So the source population was from both the blue dots and the green dots. And I just want to point out that's Quail Spring. Okay. So we took all those samples and we ran genetics from the historic samples to now. And you're going to see this crazy plot. The y-axis, so this on this side, this is just the site. So this is an abbreviation for the site, and it's also labeled in little gray font here. The region that it came from, or that it's part of. So Black Canyon is BC. That was one of the main donor sites. North Shore is blue. Littlefield is kind of its own weird place. It's extinct now. Don't worry about it. And then these triangles, these green triangles, are translocations. So that's where their frogs were not there previously, and they've reintroduced them out into the landscape. And what's cool is the further to the left on this plot, the less genetic diversity you have. And the further to the right, the more diversity you have. So we can see, hey, this population has probably the most genetic diversity. However, it's from 1998, and this population crashed. We've sampled it now, and here's the current measure. It's lost a ton of diversity, not doing so great, okay? Similarly, this one in 98, and now it's also lost diversity. It has the lowest diversity of any of the sites. And that is reflected when they go out and try to count frogs. There's not many left, right? They can actually see based on how many frogs we see, how many eggs are laid, all that stuff. The only thing that's really cool, and in a few of these cases, actually, we can see there is a gain in diversity in just a few spots. The North Shore site, this one, and this one, Rogers Spring, here actually gained a bunch. There's lots of frogs there, which is great. And what's really neat is the one site, remember Quail Spring where they mixed things up? Look, it has the most diversity. So from a management standpoint, this is an interesting conundrum because sometimes there's a lot of folks pushing against mixing things up. But in some cases, if we're thinking about like long-term persistence and the ability to weather climate change and all these other things that are happening, it might not be a bad idea if we want to really keep the species around to mix things up sometimes. So, but we wouldn't know this if we didn't have this data, which is really neat. So it's kind of like this hidden mystery behind things that really allows us to decide like, hey, we're going to prioritize, you know, obviously this site's doing really well. We're going to take some of those and maybe move them around, whatever it might be. It's a way to sort of evaluate like, how did we do? Cool. All right. So I'm going to shift gears now and talk about rivers, which is really my first love in California. And so here's a quick abridged history of rivers in California. So rivers flowed largely uninterrupted for 20,000 years after the last ice age. So in the late Pleistocene and after. Then hydraulic mining began in 1853, and it literally transformed the landscape at a geologic scale. And it was only for about 30 years. It was banned in 1884, mainly because a lot of the sediment and cobble that was being washed downstream was causing flooding down in Sacramento and the Central Valley. So farmers and others were getting kind of ticked off. Then not long after, in the 1930s, there was a huge push to basically dam and divert water and manage how we move water around California. And that included building a lot of hydropower for electricity generation. I love this image. It's sort of made up, right? But this is like a simulated satellite image from 1851. And here you can see, beautiful Sierra, some, snowy Sierras here. There's Lake Tahoe here. And what's really cool is you can see water connecting all through the Central Valley, right? This is Tule Lake, which doesn't really exist anymore, but it was huge. This is the biggest freshwater lake, I think, in North America, actually. So all this water flowing off the Sierras out into here, out into the Bay Delta. Similarly, you had lots of green water flowing out into our wetlands in here. So really cool. Obviously, this is fake. But just to give you a sense of how much water was out on the landscape, it was a lot. And just to point out how much gold mining really changed the landscape, this quote really, or this stat really caught me the first time I read it. And so now I use it all the time, but 8 times more material was excavated from the Yuba, Bear, and American watersheds, which is basically this blue square right here. Just here, 8 times more material than during the entire construction of the Panama Canal. So that's a lot of sediment and rock and dirt that is now gone from this or moved somewhere else. So that's a huge transformative change in this landscape. And then we built a bunch of dams. We still have a bunch of dams. California is fairly unique. It has more hydropower really than any other state. Almost 15% of our energy supply. That's changing, unfortunately, with these changing snowpack and reservoir levels. But it's a unique source of energy that our system is set up to provide. And with increasing need for air conditioning in the Central Valley, residential energy demands are expected to increase. and add to that that pretty much almost all of the streams in California have some sort of flow alteration, whether that's more flow or less flow. It's changed from what would be there historically. And this means that there's like the things that have evolved in those places they've evolved for thousands of years, tens of thousands of years, based on certain signals, right? Certain cues. And those environmental cues, particularly for things like salmon and frogs, really help convey really important information about when to spawn, when to migrate, right? Like when to hibernate. All those things are really important. So I'm going to show you, we have a time-lapse camera that we set up on a few different rivers. And this is the same river through a season. This is in sort of the late summer. So this is August. And you can see here's this nice cobble bar. This is the North Yuba River, by the way. Nice cobble bar. These willows are really big and bushy, right? And you can see the same time, 4 years prior, it looks a little bit different. The willows, but still pretty low flow, right? Like, so the flow is pretty low. So let's flip forward now into the fall. Well, maybe now we have our first fall rains, everything gets wet, all that organic material, all the leaves, all that stuff is falling into the river and getting flushed out, right? All the sort of dead algae, all that is really important material that gets flushed out towards sort of, used to out to wetlands, where it was then sort of energy for all the things to happen. All you need is sun. It's like a giant solar panel. And then, in the winter we get snow, and, all these things have different cues and chemistries. And so when you put a dam in or you change the flows, we're changing the hydrology, we're changing the temperature regime, we're changing the water chemistry even. And so the other thing is we're also restricting, this is a massive flood that came through in 2017. Normally this is like, well, you saw, normally like this is the level of the flow. The water is like up, at the bank, basically as high as it can go. Look, it's just wall to wall. It's crazy. And it's moving rocks. I mean, you could go and hear the boulders turning in here. So all these things have really important components, or the really important functions for the river. And so by restricting or changing any single one of them, you really can change how predictable a river is through the seasons. And so a free-flowing river on the left here has things like flood species that really need those big floods to come clear out this habitat. And then they can, they seed in here and they sprout. You have lots of native fish, all these different things. The flows can go up and down. With a dam or a regulated river, you often have a lot of non-native species. Everything is just static, right? It doesn't really change much. So oftentimes you get sort of one set of species that just sort of moves in, and they're locked in. They're really hard to remove once they get in. And so you lose sort of our natural floodplains and wetlands that were really important for all kinds of things. Turtles to spawn, frogs. This is one of my favorite pictures of Peter Moyle, who's an amazing fish biologist here at UC Davis, who's retired. Yeah, so anyway, big changes between these two things. And what that looks like in an actual hydrograph. So I'm going to show you a few plots of like what the river flow looks like. In the red is a regulated, highly regulated, what we call a hydropeaking reach. So this is hourly flows on the right, and this is sort of the average over the whole year. And so you can see over the course of the spring and summer, which this was on the right, the red line is hydropeaking up and down, up and down, up and down every day. It's like tidal almost. Versus the blue lines, these are two undammed, unregulated rivers, have this sort of winter storm period, right? And then this beautiful spring recession, we call the snowmelt recession, where all that snow is melting in the Sierras and then slowly receding down into a very stable period in the summer base flows, we'd like to call it, right? And so this is a very distinct cue that you can see, the snowmelt recession. And in a bypass, the orange lines here, it's much more reduced. In some cases, you do have a little bit of a recession, but the pattern is very different, right? So we have all these different patterns that are totally different than what was there historically. And so we can measure seasonality, which is sort of within your conditions like fall, winter, and spring, or predictability, which is how often that happens year after year after year. So how often do we have a spring recession over 10 years is predictable. And this is why that matters. So in unregulated rivers, so this is 10 plus years of an unregulated river on the North Fork American, very natural flow regime, and the frogs and salmon really cue in every year on the spring recession. They use it to spawn, to lay their eggs, because those tadpoles then emerge and have this really nice period in the summer where the algae is growing, the flows aren't bouncing around, and they can eat, be happy, metamorphose, and get out of the river, right? If you change that, and so now we have this like tidal system, and you can see that there's just no pattern whatsoever. There's like no predictability. And the problem is, if something were to lay its eggs during the same time of year it had sort of evolved to do it, those flows might drop unexpectedly, and you have now a dry rock where instead of the having a successful spawning event, you've now got lots of little dried eggs, and that's it, and it's done. And so this happens in a lot of regulated rivers, unfortunately. And we looked at this, so I can show you, this is, the cool thing is we can pull data from all these different rivers. So this is before the dams went in, and I've color coded them so you can see the ones that now do have dams and that are bypass are these black ones. The one hydropeaking one that we saw earlier that's like tidal system is this triangle. And then blue ones are still unregulated, right? And so this is gauge data that we pulled from USGS, and I calculated predictability and seasonality. And what you see is after the dam, they all shift, right? They move totally left. They lose a lot of seasonality. They lose predictability, whereas the unregulated ones still stay over here on the right. So it's pretty dramatic, right? We can see this in these dammed systems really clearly. So what the heck does that mean for our frogs, our native frogs? Well, pop quiz. Anybody in the chat that wants, or you can even just count. First part of this is I have to go out and count eggs and count frogs. So how good of a frog biologist do you think you are? How many frogs are in this picture? Anybody in the chat, go ahead and throw it in. What do you think? And then I'll show you here in a second. 22, nice. 17, okay. Can't see any, awesome. Well, so there's actually about 11 or 12 in here. And so the frogs I study, this little guy right here, they love these sort of seep kind of spring wet little areas, and there's tons of them. They all hang out, but they blend in really well. This is taken from like 6 feet away. So it's fun, but you can tell why it can be hard to work with the species when you can't find them or catch them or see them. Okay, so let's talk about the foothill yellow-legged frogs. So I'm going to skip through some of this. They've been around. They have a huge range in California and Oregon historically. Unfortunately, they're not around very much anymore. They're really cool because they're one of our only river breeding frogs, and they use a crazy wide range of habitat, from like little tiny rivers and creeks to huge rivers. And so they are, they're not doing well. They're a state listed endangered species, and they're about to be probably a federally listed endangered species. But what's really amazing is they're like little barometers of the river. And that's because they are really linked tightly with a local hydrology. You know, those river flows I was showing you, they can sort of cue in on the flow and the water temperatures and lay their eggs. And so they do a great job of hiding. They attach their eggs to these rocks. And so you have to snorkel in these Sierran rivers. Actually, my background picture is me in a dry suit, basically getting ready to go snorkel for frog eggs. And we count these eggs because one female lays 1 egg mass per year. So they're a really good indicator of population size. And I just love this picture. It's these little, you can see the cells dividing in the eggs themselves. So these are individual eggs dividing, and you can like literally see it happening. It's pretty stunning. They look like little soccer balls. Okay, so I've talked about this, they time to lay their eggs in the spring because they really like this sort of stable, low flow period, not during the winter when it's really unpredictable. The problem is when they lay their eggs in these regulated systems, you end up with this, situation where they can really desiccate or get scoured, sort of sheared off the rock and blown off. And then when that happens, that's it. And there's no survival. And so here's an example of a regulated reach where we went out and we counted eggs in the morning. And then in the afternoon, we came back when the flows had changed, and the same areas that had eggs were high and dry. And it looks like this. There's just little black specks. It's a bummer. So what I did is I went out and sampled frogs, took little mouth swabs from tons and tons and tons of frogs across the Yuba and the American River. The only thing you need to pay attention to is the blue lines is the unregulated. So I had 2 undammed, unregulated rivers, 1 hydro-peaking reach, so this red one down here, and then the rest are bypass, okay? So I was comparing the flow regimes and looking at, like, is there a difference in the genetic samples across these different flow regimes, genetic diversity. And just to also point out, if we zoom in on one watershed, these are right next to each other, right? So North Fork American, Middle Fork American, and then these watersheds actually flow into the Middle Fork. But all these little red dots are actually samples that I took from the tributaries, because there's no frogs in the main stem river anymore because of that hydro-peaking. So just so you know, the samples I'm going to talk about from the hydro-peaking reach are actually only from the little tiny tributaries to the main stem river. And what's cool, here's another one of these PCAs. And so this is across many watersheds. And so we would expect a watershed, frogs from a single watershed would probably be closely related, and they are going to cluster together, right? So inside the circles, different watersheds. So this is the North Fork American. This is all the Yuba, and they all kind of cluster together, which isn't surprising. They're all, nested in the same watershed. This is actually the hydro-peaking reach, right? So if this is 3 different rivers from the same watershed, and these 2 are 2 rivers from the same watershed, something strikes you as weird, right? Like these should be more closely related, and they're not. So if we zoom in on that a little bit, so here's our unregulated reach, and you can see just kind of a scatter. And that's a good sign. It means that the frogs are moving around a lot. There's a lot of gene flow. None of these are really that distinct from one another. Some of these might be really far away, like way upstream from the other one, and that actually is indicated in the data. But generally, good signal, right? They're mixing up a lot. Versus our regulated sites. So these are all 3 different tributaries, and you can see each of these is from a different tributary, and they're super different, really distinct. And these are the same watershed, but they're not in the hydro-peaking regime. So these are the ones upstream in the bypass. So different hydrologies, right? Different regulation regimes, and really distinct signatures. Like this is crazy different for frogs that are quite close together. And these frogs, just so you know, can move around pretty easily. They can move 7 to 10 kilometers without too much trouble, like a single individual. One other way we can look at this is using another metric of connectivity, essentially, right? And so in this next plot, I'll show you 1 is completely different and 0 is they're totally mixed up, right? So it's sort of a measure of isolation. And we usually plot this. So on the y-axis here, that's that FST metric. So, the higher the number, the more different it is. And then on the x-axis, on the horizontal, it's how far apart they are as the river flows. And we would expect, genetic theory tells us, the farther apart things are, the more genetically different they should be. So you would expect these lines to all sort of go from low to high. The farther apart, the more different they are. However, what I did not expect, and I had to rerun this a bunch of times, is that essentially the hydro-peaking reach is way up here, right? So it's like if you draw a horizontal line, it's so far different than everything else, and it's not relating to distance. It's really crazy. So the way that this stood out is shocking. because these rivers, this river in particular, has only been dammed and regulated for maybe since the late 1960s, early 1970s. That's not that long to see a genetic difference in a species, a population. These frogs take, 3 to 5 years per generation. That's pretty striking. And so, yeah, I talked about that. And then if we measure diversity, the genetic diversity, this is another way to measure it. Sites are essentially the ones on the Y here. The more diversity something has, or higher adaptive potential, it's more to the left of this dashed line, and to the right means less diversity. So there's declining population or bottlenecks. And you can see our hydropeaking sites are as far to the right as possible. And what's striking is actually most of these sites are to the right of that line, which is why they're not doing so well, and why they need help, right? So the genetic diversity generally is quite low, which could be a remnant from things like hydropeaking, hydraulic mining, who knows, lots of different things, right? But the one place where there is hope is where these sort of unregulated populations are occurring, because they can mix up a little bit more. Okay, so I'm going to wrap up, and I think just thanks for sticking with me. I know it's a lot of stuff I'm throwing at you, but I just want to say that these sort of genetic tools are really powerful, and they're a really amazing way to sort of inform actual management for whatever it might be used. And that diversity is something that's like, we read a lot about it and we talk a lot about it, but it's rarely actually used in conservation management. And it's not very considered for long-term persistence in a way that it probably could be. So I think it's a really neat tool to sort of leverage amidst all the other stuff. Because I mean, my favorite part is to go out and survey and snorkel and catch animals, but sometimes we also need to use other tools as well. And I think the other part of this is, working across disciplines. I've learned a ton about hydrology and geology that I never would have learned. And I think sort of working across these different disciplines is really helpful. And we need more of that to really sort of have better efficacy in how we do stuff. So I will leave it at that and say thanks. This is my daughter in a bucket, my little tadpole. Yeah, I'm open to questions. I'd love to hear what folks think.
Thank you for your presentation, Ryan. It's really fascinating, all of the information that's available now to try to make better conservation and management decisions. We have some questions in the chat. The first one that came through was when you were talking about sort of the genetic diversity plots. Do you suspect that diversity would change over time in the long term after translocation?
Great question. Yeah, I mean, I think that's the hope, right? If we can introduce enough, if there's enough genetic diversity and there's enough gene flow between populations, we would expect it at minimum for it to be stable, right? Sort of the line to sort of be stable and not be losing more. But even more, like we would expect potentially to sort of increase to a certain point. It's not going to just keep increasing, but if we can get it up to a point where that indicates the population size is actually bigger, it actually tells us the more genetic diversity you have, often the larger the population. So it can tell us kind of both, but these things take time, right? So sometimes we might need to go back out in 5 years or 10 years. And the problem is a lot of the conservation plans don't really build that in currently. So there's more discussions about that and the importance of it. Yeah, good question.
And then there's a question from Melanie. She's interested in hearing more about zooplankton and its nutrition for salmon. And she asks, can you lead the salmon to the zooplankton?
Yeah, it's a great question. And there is a whole food for fish program with Caltrout, actually, that I'm working with them and rice farmers and a lot of other folks from the ag side of things, because it turns out that a lot of the agricultural fields that when they flood, particularly for rice, but even for other stuff, if it's not being used, those sort of floodplain, artificial floodplains, we'll call them, right? They're just sort of croplands that have been flooded, are just massive generators of food, right? It's like a solar panel. You just add water and sun and energy. And what's cool is if we can connect those areas, so if we can sort of get the water that's on those fields to drain into the river or into places where the fish actually can access that food, then yeah, we are sort of leading the fish to the food in a sense, right? Alternatively, we're really trying to figure out ways to get the fish onto the few floodplains that exist. So, but that requires water, which is, obviously a hot topic in many ways. But yeah, great question.
And then we just had some appreciation for you in the chat, people saying that it was fascinating and they really appreciated your data visualizations. There was a question that I asked for clarification for, but it was about different breeding seasons. So I'm waiting to hear back.
Oh, like salmon? Frogs?
She said the question was for frogs.
Yeah, so for frogs, at least for the species I talked about, for foothill yellow-legged frogs, they're a spring breeding species, and they really cue in on that sort of spring recession period. What's fascinating is the same population, say like in one of the rivers I study regularly that I've been monitoring, a year like this year where we're in like one of the probably driest years we've had, I'm seeing breeding starting, now or a few weeks ago, even really early. But in a really wet year, the same frogs, same population can delay their breeding and start as late as like late June, early July. So it's very plastic in their ability to sort of flexibly move to the right cue, mainly because the flow, they're cueing on the temperature and the flow in a way that's like sort of pretty amazing, right? So I think, yeah, but they only breed sort of that, they need those cues and because they only use it once per year.
Thank you. That response got a wow from the person who asked that question. Robert had a comment that it would be interesting to use these kinds of tools to determine the native biodiversity in the Pajaro River system here in our area. I don't know if you know of anybody doing work like that around here.
I think I do know some folks. I also work on, I didn't talk about it at all, but environmental DNA, which is where we take water samples and we extract DNA out of the water. And then we can sequence that and see what organisms the DNA belongs to. So it's kind of a quick way to get a count of what's in there. And so I believe there were some folks that were doing some eDNA sampling on the Pajaro. I need to double check that, but yeah, no, absolutely. I mean, it's kind of amazing the tools that exist. And I think it's important to remember not to get too tied down to any one tool because they're all developing quickly and like pick the best one for your question. But absolutely, no, I think there's a lot of room to leverage these new tools.
And we have another question from Inger Marie about, can you talk a bit about the foothill yellow-legged frog in Big Sur area and why they are not there?
Yeah, historically they used to be much more present along the coast in a lot of the coastal systems, and I would say, we're still trying to understand exactly why. It's probably a combination of a lot of things. So chytrid is a major factor for a lot of amphibians, fungus basically that kills them. Invasive species like bullfrogs, which are everywhere, love to eat tadpoles and even adults. So bullfrogs have certainly had a huge impact, as well as probably some other invasive fish species that eat tadpoles. Plus all the other things as far as water diversion, sedimentation, like lots of things that we probably don't have as a great handle on. So it's kind of like death by 1,000 cuts, although some of those cuts are much bigger than others. But yeah, I think that's there's a few spots where they might be able to be reintroduced, but it's hard because you don't want to put them in a place that they're just going to sort of disappear from again.
And then Ryan, there's a question. Is there anything that we can do, practically speaking, to support the frogs or stop activities that are causing their decline?
That's a great question. I mean, honestly, I think just being aware that they exist in a lot of places, even if you don't see them, and, you know, get people fired up about even like the, you know, your local native chorus frogs or tree frogs that you might have. Like, they tell us stuff, right? They're an important part of the food web. They're important in many ways. And I think just sort of having them on the radar, I think, is great because honestly, as somebody who's been doing research on frogs for a long time now, 20 plus years, it's kind of an uphill battle in the sense that most of the energy and attention really goes to, and I do stuff with salmon too, but most of it really goes to salmon, right, in California for water reasons. And so making sure that we're thinking not just about one thing, but all the things is great. And so thinking about that, but yeah, get your kids involved, get them out. And the more that, you know, folks, I think, appreciate that they're nice to have around and they will be missed is, I would say, a good first start.
So I don't see any more questions in the chat. If you have one, please put it in there. I had a question. And you know, you talked, Ryan, about how much water in California is used for energy generation. So there's obviously a balance between energy and conservation, but what, you know, having the data that you have and the knowledge that you have, what would you like to see in terms of changes that could be made, like science-driven changes that could be made in terms of how we manage water in California?
Yeah, I think that's a great question. And I think I'm working with a bunch of folks that are actively involved in that same concept. And it relates to, you know, the reality is a lot of these dams are not going to come out, you know, like they're going to be there. It's pretty hard to manage that. However, there's places where I think, for example, that hydro peaking reach, there's not much we can do other than change the flow regime. That would help. But we also, you know, in some of those places, that's not an option. So I would not prioritize that place. I would prioritize other places where maybe a bypass reach, right? Like where we have more ability to sort of pull the levers or turn the knobs to manage the flow. And honestly, the biggest push right now is to manage for functional flows, which is sort of all those things I was talking about, you know, the winter storms, the spring recession, the low summer flows, like all of those things that happen through the year provide really important functions to the ecosystem. And so even in regulated systems that have dams, we can still sort of mimic some of those things, even with less water than we historically did, but you can sort of put some of that function back into the river. And the more we can do that, and there's trade-offs to that, certainly, but the more we can do that, I think the better chance we have at sort of keeping these systems intact in a way that makes them, you know, feasible to do things or keep them intact for the future. So I think, to me, that's exciting, and it's a nice it's a nice way to balance a lot of the needs that exist, you know, in a lot of these systems for water use. So yeah, it's sort of function of the flow, not just sort of, it's not like more water or less water or changing it, but it's like, what is the actual, what can we do to actually make it look more like it did historically? Those patterns are important.
Is there any watershed area that you would praise as doing a good job at that yet?
I mean, so I've been working a lot with like the federal relicensing program, you know, a lot of different rivers that have to be relicensed for hydropower. And it's now written in a lot of them that they have to avoid certain flows in the spring because of frogs or because of fish. And so I think it is becoming more accepted in that way. I think there's some cool work going down right now in LA, where actually they're looking at sort of managing these like highly altered systems, but basically how can we put function back into those systems, which is really exciting. So it's right now, it's honestly just sort of getting started. And there's a few case studies up on the Eel River, South Fork Eel, I think is one. And then we're actually working with some folks in the Little Shasta River to sort of think about, you know, like how can we manage the flows potentially, or like what would that look like? So yeah, there's a few little case studies where we can start playing with things and thinking about it. But yeah, it's a slow process.
There are a couple more questions here now. Diana asked, you could talk a little bit about why you were surprised that there was more diversity when the populations were mixed. And now you were talking about sometimes scientists try to avoid mixing for adverse.
Yeah, I mean, I think sometimes we don't necessarily know, like there's not many examples of that, because in some cases you can actually have, there's so little diversity that even if you mix them, they're sort of inbreeding happens, right? And so you can actually have the opposite. Like if you mix two populations that are really locally adapted, you can actually sort of make the opposite happen, where it's like deleterious, essentially, where you can have like mutations that are bad, and then the population crashes and dies, essentially. So we don't necessarily know that like just mixing two different things is going to work out sometimes. There's lots of things under the hood that can happen with that. So it is pretty cool that in this case, and it does sort of correlate with what we're seeing. The population is large and doing really well, but it's not, it's not going to be the end all be all for everything. But yeah, I was surprised that it was as strong signal as it seems to be.
And then Michael asks, are you optimistic that frogs will survive the current global warming situation?
I don't know. I actually had a really interesting interview with some folks from Sierra Magazine, I think, an article that they're writing about frogs and climate change and climate refugia that's coming out here in a month or two, which should be interesting. But I think in some places there's hope, and in other places I don't have much hope. So I'm trying to find the places where I can like invest energy where there's a chance that we can actually do stuff.
And then Diana had a question about what zoos are helping to keep frog populations alive here in California.
Yeah, the Oakland Zoo, actually, there's a really cool project with the Oakland Zoo. They're working with a couple, actually 2 or 3 different species: California red-legged frogs, Sierra yellow-legged frogs, mountain yellow-legged frogs, and foothill yellow-legged frogs. All of which are sort of our native big frog species. But yeah, they've been rearing frogs at the Oakland Zoo, and then we've been releasing them, or sort of head starting them out into the North Feather River, which is one of these sort of highly dammed, highly altered rivers. And it's been pretty cool to see for now 2 or 3 years running. They just released a new batch of frogs out last week, I think, to sort of try to get the population built up again, see what will happen. And I think LA is also working, has some frog breeding populations down there too.
Okay, and then the last question that I see is your thoughts about Klamath River dam removal.
Just throw the grenade in there right at the end. Yeah, I think, yeah, I mean, ultimately, I think that, but that's a whole separate sort of set of things, right? That's enmeshed in a lot of the different parts of history of California in many ways, right? But I would say overall, yeah, I think the dam removal in that case is going to be really, it's been a long, it's still a long contested process, right? But I think at the end of the day, those types of projects are going to be the kinds of things that we need to do. Not just removal, but that magnitude of things, I think, is going to be something that we need to be working at if we really want to have a chance at saving some of these systems or combating, the impacts of climate change. And I work with folks on the Klamath, and there's folks that have been trying to work towards that for 20 years. And it's been a long battle. And I think on both sides to try to come to sort of an accord. And I think the reality is that we need to figure out ways to work together and across these sort of disciplines. And I think I'm excited about the ability to take some of these new tools as ways to give us more information to make, just more informed decisions about what to do. Because I think in many cases, we don't know in some cases, like what the impacts will be of climate change and what's going to happen if we take out a dam here or do this. So we need research about that, and we need to sort of have a good sense. We also need to be willing to embrace that change, right? Like we can't just not do anything. So sometimes you got to kind of get into it and see what happens. And, you know, it's not going to be any more broken than it already is in many cases.
Okay. Well, thank you so much, Ryan. There was a lot of appreciation in the chat for your interesting presentation. Thank you so much for your time tonight. And thank you to everyone who joined us. We will be continuing to have some online, for now, Zoom-based virtual speaker series. So we hope you can join us again in the future. Good night, everybody. Thank you.
Thanks all.